Alright, this video is going to take you through the steps involved in how a DNA replicates itself. So remember when we talked about the cell cycle and we talked about the S phase of the cell cycle and how the DNA makes an exact copy of itself during this particular stage of the cell cycle so that the new cells that are made, every cell has our exact genetic information in it. We want to keep passing on this exact genetic information to the new cells that we're making. And so this is how this process works. It's actually remarkably simple for such a complex and such an important process. So we're going to start with the DNA strand. Remember, we got two strands of DNA, double-stranded. The backbone, the black, would be the sugar, the ribose, and the phosphate group. And then we have these nitrogen bases that are holding the two strands together. So A's join with T's, C's with G's, just like in the last lecture video. So the first thing that has to happen in this process, and I'm going to be oversimplifying this a little bit, but the two strands of DNA need to unzip and unwind. And this is done by a chemical or a molecule that's called DNA helicase. Basically, DNA helicase is going to come in here and it's going to break these two things apart so that we kind of get two linear strands here. All right, so if we follow this strand all the way down, okay, here's the A, C, keep going, T, C, follow the strand, G, T. All right, and then this strand over here, which would be this strand right here, okay, so this would be T, G, A, G, C, A. All right, so this strand here followed this strand, and this strand here followed that strand. Okay, so the DNA helicase comes in and it unzips and it unwinds the DNA. It separates this. Now, it doesn't actually do the entire strand of DNA. It would take way too long. So it's actually doing this in different sections all along the DNA at the same time. And the point at which it starts to unzip and unwind itself, we call that a replication fork. We don't need to get into all of the super duper process of that. Just know that the DNA doesn't completely do this all in one swoop. It's doing it all over the place in different parts of the DNA. So now that we have the DNA or sections of the DNA unzipped and unwound, the next step is that we're going to basically insert two new strands of DNA in between the old or the original strands. Okay, so here's the original strand. Here is an original strand. So now a chemical, an enzyme called DNA polymerase, is going to come in. It's going to insert two new strands right in between the original ones. And it's going to throw in the base pair that's needed to match up. Okay, so since this is an A, this has to be a T. Since this is a C, it has to be a G. So it's going to come in here and it's going to insert the strands and the matching nitrogen base. Okay, because these are floating all over, so it knows where it needs to throw it in. And now you can see we have two molecules of DNA. Okay, here's one molecule of DNA. Circle it kind of. Okay, here's the other molecule of DNA. And if you look, they are identical. Adenine, thymine, adenine, thymine, cytosine, guanine, cytosine, guanine. Okay, T-A, T-A. Each of the new molecules has one of the original strands from this, what we started with, and it has one new strand. Now, last thing that has to occur. This process is so important. We need to make sure that the letters, that there's been no mistake in the letters. Okay? So the DNA polymerase, it does a lot of different things, but the last thing that it does is it kind of goes through here and it proofreads. All right, if for some reason a T was put right here, it would be like, wrong, I need to go back, I need to fix that, and it'll put in the proper nitrogen base. So it will go through and it'll proofread the 2 new strands of DNA, and then when it's done, it's basically going to zip itself back up into that helix shape. And now we will end up with 2 new molecules, genetically identical molecules of DNA.